Hi there, my name is Martin and I want to tell you about new software which can change the way immunologists analyse their data. Modern lab instruments are producing a deluge of high quality data that often goes underutilised because immunologists don't have the computer programming skills to take advantage of it. In many institutions, bioinformaticians are outnumbered by their non-coding colleagues, even as many as 20 to 1. What's more, instrument readouts from different manufacturers are difficult to integrate with each other, and software is typically focused on one type of readout. This hampers immunologists from getting the overview they want from their data. Tercen solves these problems by placing a user-friendly layer over powerful statistical analysis packages such as R or Python. This allows a non-coder to build sophisticated data models by dragging and dropping elements on a screen while Tercen produces visualizations on the fly. Tercen simplifies the data wrangling and file uploading, and because it's cloud-based, it makes even worldwide collaboration so much easier. It even helps bioinformaticians optimize their effectiveness. So much of their time is spent performing admin tasks like adjusting graphs. Now they can focus on designing better analysis techniques and then plug their code into Tercen for the team to use. So how does this work for immunologists? Let's look at some examples of the kind of data analysis that can be done using Tercen, such as analysing high-dimensional data from a flow cytometer. The core of a Tercen analysis is the workflow. It's a logical progression of data from uploading to transformation to visualization, and workflows branch as different techniques are explored. Here we've tried both TISNI and UMAP to see which dimension reduction technique gives us a better result with our flow cytometry data. In this workflow, we've added the meta-annotations that accompany the patient samples, and by making this relationship, we can enrich the information shown by the visual. For this case, patient samples have been sorted into subpopulations on a plate, and these cells are sequenced. The challenge at this point is working out how to integrate the sequencing data and the FACS sorting data. In this workflow, Tercen has integrated the FACS, plate annotation and sequence data. Calculations and visuals are now available on the integrated datasets. For example, because Tercen can colour by any element, we can make multiple visualizations of the same graph to identify correlations. Tercen also has ways to help immunologists engage in translational research. Going from bench to bed can involve many different technology platforms, such as flow cytometry, cytokines, and other single-cell omics. There are also many different additional sources of information that provide insight to the readout data. These can be patient markers from hematology, clinical annotations from drug treatments, or meta-annotation about patient demographics. All of these need to be integrated before the researcher can ask the right questions of the data. This workflow analyses a clinical trial investigating vitamin D use in the treatment of allergies to birch pollen. Here, flow data, cytokine data, and proliferation data are combined in one table. Annotations for sample, patient, and marker are integrated. Now the immunologist can get an overview of all their data in one graphic. So, hopefully this video has given you a brief look at how you can transform your data analysis. We would like to encourage you to sign up and see for yourself. Tercen is free for the individual user, so there's really nothing to lose. And there are many more great features that I haven't covered here. Book a session with us if you'd like some in-depth help, because there's nothing we love more than talking about data. Thanks for watching.